So welcome all participant in another video tutorial from Genome Biotech, and as always I am your host Dr. Nitin Wahi, and we are going to start from today's topic of talk or discussion would be the structure of proteins. So we are going to talk about the structure of proteins. What is protein? What are the different components of the protein? How the structure of the protein is being oriented. Now, see, when you're talking about Pathfinder, in Pathfinder, you see that's a basic book. You see that there are four different orientation for the structure of proteins. The simple orientation is the linear structure of protein. Okay, when you're going to study about the linear structure of proteins, then see this. What is written here? Proteins have four level of structural organization. Which are the four proper level of protein structural organization? The four proper level of protein structural organization are the primary structure. The primary structure consists of a polypeptide that is made up of amino acid sequences linked together with each other. Okay, and this is referred to as the primary structure of proteins. The amino acids are connected by peptide bonds. The primary structure of the polypeptide is determining the higher level of structural organization in the case of proteins. So in the case of proteins, what is going to happen? Proteins consist of linear array of amino acid residue joined together with the help of peptide bond. So it consists of linear array of amino acid residue, and those linear array of amino acid residue are joined together with the help of peptide bond. So they are the one which will be joined together with the help of peptide bond. Okay. So what we have to study from here is that we will have to study about the structure of protein. Now, how we have to study about the structure of protein? The primary structure just made up of linear array of amino acid residue joined together with the help of peptide bond. Primary structure में क्या होता है? Linear array of amino acid residue joined together with the help of peptide bond. Linear array of amino acid residue joined together with the help of peptide bond. With the help of peptide bond. So there are peptide bonds which are present, and these peptide bonds are joining the amino acid residues together with each other. For example, there is an amino acid, then there is a peptide bond, then there is an amino acid, then there is a peptide bond, and so on. So when there is this type of an arrangement, then it is referred to as the primary structure of protein. Okay, the primary structure of polypeptide is just being made up of the amino acid sequences, and these amino acid sequences are being joined with regard to each other. Okay. Now, what we have to study about is the secondary structure. Now, primary structure, the predominant bond is the peptide bond. Now, the polypeptide which is present in the case of protein is going to fold over itself, and when the polypeptide is going to fold over itself, then it will form a structure that is referred to as the secondary structure of protein. Now polypeptide is going to fold over itself, then it will produce another structure, and that structure will be referred to as the secondary structure of the protein. Now we have to see that how these polypeptides are going to fold over itself. Okay, so that we are going to see from Lippincott. Okay, I'm sharing Lippincott with you all. Now this is Lippincott. Probably you would have you are going to visualize this particular screen, and now see there are 20 amino acids that are going to linearly form the structure of the protein. Now if these are 20 amino acids that will form the structure of the protein, what we have to study here is they are being joined together by the peptide bond. So this makes the primary structure of the protein. So you look here, this is the primary structure of the protein. You can look at focus on this particular diagram, that how the structure organization of the protein is based. One is the primary structure. Primary structure consists of amino acid residues, and these amino acid residues are being joined together with the help of peptide bond. Then it is referred to as primary structure. Now protein primary structure is going to fold over itself. As the protein primary structure is going to fold over itself and lead to the formation of hydrogen bonds that you can visualize. You can find that there are hydrogen bonds which are present, and these hydrogen bonds which are present here, they will lead to the formation of another hierarchy of protein structure, and that is referred to as the secondary structure of protein. Okay, then this protein is also further going to fold over itself. As the protein is further going to fold over itself, it will form another hierarchy structure, and that is referred to as the tertiary structure of protein. When large number of tertiary structures are going to be combined, they will form the quaternary structure of protein. See, primary structure is just made up of amino acid sequences which are linearly attached to each other. Then primary structure, after the primary structure, once the hydrogen bonds are being produced in between the amino groups and the carboxylic group CO between the electronegative element and an electropositive one, then these bonds which are being produced, the hydrogen and the hydrogen bonds which are being produced, will lead to the formation of secondary structure, when the protein is going to fold over itself and forms another hierarchy structure that is referred to as tertiary structure. When large number of protein molecules are going to be joined together, it will lead to the formation of another structure that is referred to as the quaternary structure of protein. So there are four different structural organization: the primary structure, the secondary, the tertiary, and the quaternary structure of protein. So these are the four different types of organization that the proteins have. Okay. Now what we have to study here is that that will form the secondary structure, the tertiary, and the quaternary structure of protein. Now, primary structure of protein. What is the primary structure of protein? How the primary structure of protein is being produced? Now, primary structure of protein. In protein, amino acids are joined covalently by peptide bond, which are amide linkages between the alpha carbonyl carbon atom of one amino acid and the alpha amino group of another amino acid. So, what happens in the case of proteins? In the case of proteins, there are amide linkages which are being produced. Now, how these amide linkages are being produced? Amide linkages are being produced in between the alpha carbonyl group of one amino acid and the amino group of another amino acid. When both the alpha carbonyl carbon of one amino acid and the amino group of another amino acids are going to be linked, then it will lead to the formation of a peptide bond. For example, valine and alanine can form the dipeptide vinyl alanine. For example, valine and alanine are two different amino acids. They are being joined together with each other and they are forming vinyl alanine through the formation of a peptide bond. Okay. So how the primary structure of protein is being oriented? Just by joining of amino acids together with each other. Now let us look at this biochemical reaction, that how does the primary structure of protein is being produced. You look here, there is the first amino acid valine, there is the second amino acid alanine, and both the amino acids are reacting together with each other. When both the amino acids are being reacting together with each other, there the carboxylic group is going to react with the amino group, and the carboxylic group and amino group are going to react with each other, and then it will lead to the formation of valine alanine. This bond which is being formed is referred to as the peptide bond. The CONH bond is referred to as the peptide bond. So just simple, it is a reaction. The peptide bond involves the formation of a peptide bond in between the alpha carbonyl carbon atom of one amino acid and the alpha amino atom of another amino acid in a nucleophilic acid substitution reaction. I have already told you the definition about it. Okay, so this is how the reaction takes place. इस तरीके से रिएक्शन होता है और आखिरी में बन जाता है विनाइल एलानीन. Characteristics of the peptide bond. There are two different characteristics of the peptide bond. I have already told you that the peptide bonds, the omega bond, can have two different conformations. One, it could have a cis conformation, another it could have a trans conformation. Now, where is the cis conformation? Where is the trans conformation? If you have any query, you can write in the chat box. Okay. Now, what we have to study here is that there are two different conformations of the peptide bonds which are present. Now, which are the two different conformations of the peptide bond? One is referred to as omega equal to 0 in the case of cis and omega equals to 180 degree in the case of trans. This is how the two different orientations are present. Okay, so these are the two different orientations which are there. Next, what we have to study is that this forms the primary structure of the protein. This forms the primary structure of the protein. And now we have to look at the secondary structure of the protein. Okay, so trans is much more stable as compared to the cis. That's the same we have studied in the case of Pathfinder. In the yesterday's lecture, I have already told you what is the value of omega, how the value of omega is being oriented, what is psi, what is phi, how these molecules are being oriented with regard to each other. That we have to look forward. Okay, now please, we are going to move forward. This is it, this is it, this is the primary structure. Now we have to look towards, we have to look at the secondary structure of protein. That what is the secondary structure of protein, and that we have to see that the primary conformation of the protein or the primary structure of the protein can fold over itself. I'm taking a annotator to describe this particular thing. For example, there is the primary structure of protein. Okay, now where is the primary structure of protein? Consists of amino acid, and amino acid, and this there are n number of amino acids which are being joined together with each other. Okay, so there are, I'm taking this figure, there are n amino acid residues which are being joined with regard to each other. There are many number of amino acid residues that are being joined with regard to each other, and towards one end there is the amino group, and towards another end there is the carboxyl group. There is an amino group at one end, and there is a carboxyl group at the other end. There is the carboxyl group, COOH group. Okay, so this is the carboxyl group at the other end. Now this amino acid, this is referred to as the primary structure. This is referred to as the primary structure. This primary structure is going to fold over itself. When this primary structure is going to fold over itself, then it will lead to the formation of another structure that would be referred to as the secondary structure. Now this primary structure has folded over itself. As the primary structure has folded over itself, because how this primary structure is able to fold? Because the carboxyl group, COOH group, this oxygen was making a hydrogen bond with hydrogen atom from the amino group, and because of all these types of different types of bonding which was taking place there, the secondary structures are being produced. So how the secondary structures are being produced? Secondary structures are being produced in this particular manner. Proteins also makes helical conformations because of hydrogen bonding in between the adjacent amino acid residue. The hydrogen bonding takes place in between adjacent amino acid residue, and that is how it leads to the formation of such structures. Okay, now moving forward, there are different conformations which are present. One is referred to as the alpha helix, another one is referred to as the beta plated sheet structure. Now what is this alpha helix that we have to study? Let us first study with Lippincott, because there are good diagrams which are given in Lippincott. Okay, now see, this is the Manhattan test which is being given, how the colors are being produced. You can look at it in great detail. Okay, I'm moving to the alpha helix. Now see this. No, this is not about the alpha helix. Here it is, the alpha helix. Now see what is going to happen. The polypeptide, how the secondary structure of proteins are being produced. The polypeptide backbone does not assume a random three-dimensional structure, but instead generally form regularly arrangement of amino acids that are located near to each other in a linear sequence. These arrangements are termed the secondary structure of the polypeptide. Okay, there is a localized arrangement of amino acid residue, but instead generally forms regular arrangement of amino acid residue that are located near to each other in the linear sequence. These arrangements are referred to as the secondary structure of polypeptide. Now let me just make you understand what this is going to be. Now supposingly there is, you are going to your school, and what happens in the school? The first thing that happens in the school is a prayer. Every class has n number of students, and all are standing in a particular line. Okay, now what happens? Now the prayer gets over, and now you are going to move towards your classes. So everyone has its seat fixed, and everyone want to get seated along with their friends, ताकि time pass अच्छा हो जाए, so that you can pass the time during the entire lectures. That arrangement is on the basis of hydrogen bonding, that which molecule is being bonded with which molecule, and that is how the orientation takes place, that how which molecule is going to be bounded with which molecule, and it will lead to the formation of different types of structures. This is what happens in the case of protein. In the case of protein also, firstly, the amino acid sequences were there. The protein was a linear molecule. Now what has happened? Now the localized interactions have taken place. Because of localized interaction, the protein molecule, although was linear, but now because of localized interaction, see this, what I'm trying to draw is there are interconnections which have been made, and now the protein has gotten a folded structure. The folded structure of protein formed due to hydrogen bonding is referred to as the secondary structure. Simple. What is secondary structure of protein? The secondary structure of the protein is a folded structure of protein formed by hydrogen bonding in between the amino group and the carboxylic group present within the polypeptide backbone. So the polypeptide backbone amino and carboxylic group binds together with each other. As they bind together with each other, this will lead to the formation of a structure, and that structure will lead to the production of the secondary structure. Okay, so secondary structure is being produced because of this binding or because of this folding. Okay, now we are going to move forward. Now, first secondary structure that we have to study is alpha helix. Then we will be talking about beta sheet. Then we will be talking about beta bends. Okay. The first is referred to as first and the foremost structure that is present is alpha helix. Now, alpha helix can be categorized into two different types: the right-handed alpha helix as well as the left-handed alpha helix. Now, what happens in the case of right-handed alpha helix that we have to see? What happens in the case of left-handed alpha helix that we will have to see? Okay. So what happens here is that you can look forward. See this. This is what I was trying to explain it to you. When the polypeptide is present in its linear form, then the carboxylic group of one amino acid began to interact with the amino group of another amino acid. And there are hydrogen bonds which are being produced. That is why the entire polypeptide structure is being altered. Okay? So what is going to happen in the class? Okay? Supposingly the class began to take place. There was a class of say graduation. Okay, graduation first year. Everybody is there in the class for the first time, and they are sitting in accordance to their roll number. A strict teacher used to come in the class and says that make the line according to your roll number. Okay, students. So every student is making the line in according to their roll number. Now the class has passed. Now there is the time of farewell. Now what happens in the case of farewell? Or dude, there is any teacher who says make a line in terms of your roll number? No, there are arrangement. Each and every bond is being produced. There are so many different types of bonds which are being made in the class. You all know very well as compared to me which type of bonds I'm talking about. Okay, so there are so many hidden bonds which have been produced, although everybody knows about it, but nobody is talking about it. Okay, so there are so many different types of bonds which are being produced, and those are referred to as hydrogen bonds in between the amino group and the carboxylic group. And because of those amino bonds, because of those hydrogen bonds, there can be intra-chain hydrogen bonds. The entire structure of the polypeptide is being altered into a helical conformation. So this is the helical conformation, the structure which has been produced. Okay, side chain of the amino acid are always extended towards outside so as to make the polypeptide highly stable. Now let us look what is given in Pathfinder, then we will go into detail with this Lippincott. Okay, so please, I'm just switching back to Pathfinder. You remember this particular structure because I will be talking about this particular structure for a number of times in this alpha helix. Now see this. Alpha helix is a rigid rod-like structure that forms a polypeptide chain folded into a helical conformation. It is a rigid rod-like structure. This is a rod-like structure. When you elongate it out, okay, this will form a rod-like structure. It's a rigid rod-like structure which is being formed when the polypeptide chain elongates in order to form a helical conformation. You look here, it is looking like a rod-like structure. Okay, this is the rod-like structure. It is a rigid rod-like structure formed when the polypeptide folds itself into a helical conformation. There is the polypeptide folding itself into a helical conformation. I'm trying to make that helix again. So this is how this polypeptide is being arranged. It is being arranged in this particular helix. There is the amino group, NH2 group, and H2 group. NH2 group, and there is another group that is present, the carboxyl group, COOH. Okay, there would be the COOH carboxyl group that would be present. Okay, so this is how the arrangement of amino acid takes place. Moving back to Pathfinder, just a minute. Okay, sorry for all these. The screw sense of the alpha helix can be of two different types: the clockwise and the left-handed. The right-handed, that is clockwise alpha helix, and another one would be referred to as the left-handed, the counterclockwise alpha helix. So I have to underline this out because I will be explaining you certain things from this particular part. What is this clockwise and what is this anti-clockwise alpha helix? Okay, so we are going to talk about alpha helix. Alpha helix is having two different orientation. One is referred to as clockwise, another one is referred to as anti-clockwise. Now how can we understand this out? Please look here, I'm making a diagram to make you all understand what I am talking about. Okay, and now please look here. See, we are going to classify the alpha helix into two different types: the right-handed alpha helix, and another one would be referred to as the left-handed alpha helix. I would be drawing the alpha helix in front of you. Okay, there I'm going to draw the alpha helix. Alpha helix. Okay, there would be the alpha helix. Just a minute. Now alpha helix can be classified into right-handed as well as left-handed. So I'm going to draw both the right-handed as well as the left-handed in front of you, that what is this right-handed and what is this left-handed alpha helix. I'm finding the different tools to draw. Okay, in the case of right-handed alpha helix, the direction of movement of the helix would be in right-handed direction. See, this is referred to as the right-handed alpha helix. When you look towards the left-handed alpha helix, this is referred to as the anti-clockwise left-handed alpha helix. This is referred to as the anti-clockwise left-handed alpha helix. This is right-handed alpha helix. This is left-handed alpha helix. Okay, so I'm just writing it down. Right-handed alpha helix. This is right-handed. I'm finding difficulty while writing with this mouse. This is right-handed. Okay, and there is the other one that is referred to as the left-handed alpha helix. It's writing here, left-handed. Okay, so there you find the left-handed and the right-handed alpha helix, which is much more stable. Right-handed alpha helix is said to be much more stable as compared to the left-handed alpha helix, because right-handed alpha helix will have much more region occupancy in the case of Ramachandran plot. In the case of Ramachandran plot, right-handed alpha helix will be much more occupied as compared to the left-handed alpha helix. Okay, now see what is going to happen here is that I'm just clearing all the drawing. See why this right-handed alpha helix is much more stable. Now, do you know which type of amino acids are present in us in the case of humans? In the case of humans, the alpha amino acid, in the case of humans, L amino acids and D sugars are present in higher eukaryotes. In higher eukaryotes, just a minute, I'm taking another thing so that it could remain written. See that we have to write. In higher eukaryotes, in case of higher eukaryotes, note, in higher eukaryotes, in higher eukaryotes, left-handed or L amino acids and D sugars are present. Now see this, when there are L amino acid, then how can you make an alpha helix? You can make a right-handed alpha helix. I have already told you that L amino acids will make right-handed alpha helix and D amino acid will make more stable left-handed alpha helix. See, in my last lecture I told you, if something is left-handedly coiled, then it can be coiled over itself only in right-handed direction. If something is right-handedly coiled, then it can be coiled over itself in left-handed direction. I gave you the example of ponytail in my last class just to explain this particular point. So right-handed alpha helix is much more stable in the case of humans and is found in much more large number in the case of humans. Why? Because the amino acids which are found in humans is left-handed. It is levorotatory. That is why right-handed alpha helix are found much more. Next, we would be talking about this particular alpha helix, that you can visualize there is the alpha helix that is present. Okay, now, please just give me a second so that I can remove all these annotations. I'm clearing all the drawings. Now, this is the alpha helix that you have seen. So, hydrogen bonds, and alpha helix is stabilized by extensive hydrogen bonding. So, again, back to Pathfinder, the most important bond which is present in the secondary structure of the protein is actually the hydrogen bond. See what is written, stabilized by hydrogen bonding. This question has been asked again and again, very large number of times. So the most important bond is hydrogen bond. So there are the hydrogen bonds which stabilize the secondary structure of protein. Where are the hydrogen bonds produced? In between the carbonyl and the amino groups. So the carbonyl and the amino groups are going to react together, and when these molecules are going to react together, then it will lead to the formation of hydrogen bonds. So here it is. So these will be the molecules that will be reacting together, and these will lead to the formation of hydrogen bonds. Now I've told you what is right-handed alpha helix, what is left-handed alpha helix, which is much more stable, right-handed. Now right-handed alpha helix are energetically more favored because we have L amino acid residues in our body. Okay, now see this. This is very important. There are 3.6 amino acid residues per turn of the helix. Okay, how many number of amino acid residues are present in the helix? 3.6 number of amino acid residues are present in the helix. See this. There are 3.6 number of amino acid residue per turn of the helix, and the pitch of the helix is 0.54. Again, there are two questions which have been asked from this particular part. Now, what do you mean by the pitch of the helix? What is this written 3.6 number of amino acid residue per helix? When we are going to talk about here, we are going to talk about 3.613 alpha helix. We are talking about this particular alpha helix. What alpha helix we are going to talk about? 3.613 alpha helix. I'm just pointing this out. We are only going to talk about this particular alpha helix. Now, alpha helix can occur in several different orientations. I am drawing an alpha helix in front of you. For example, if I am drawing this particular alpha helix in front of you, okay, so I have used a pencil and tried to draw alpha helix. Then in a particular turn that you can visualize here, in this particular turn, how many number of amino acid residues are present? Do you know? 3.6 number of amino acid residues are present. 3.6 number of amino acid residue. 3.6 amino acid residue per helix. Okay, this is referred to as the alpha helix. 3.613 alpha helix, because it's one helix contains 3.6 amino acid residue. Why it is referred to as 3.613 alpha helix? Now you might be thinking why there is the word 13 that has been used. The word 13 has been used because of a particular reason. Now you see that how many number of atoms are involved in the hydrogen bond. For example, you can look at this particular structure. In this structure, what you are going to find is that there is the carbon, there is an oxygen molecule. Now this oxygen is going to bind with this hydrogen molecule, okay, this hydrogen atom. Now count the number of atoms present in between them. Now 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So these are 13 atoms which are given. If you are looking at the new Pathfinder, there sir has clearly illustrated writing the number of atoms which are present from 1 to 13. And because there are 13 atoms which are involved in the formation of hydrogen bond, that is why the name is 3.613 alpha helix. The name is 3.613 alpha helix. So this is very important that the name is 3.613 alpha helix, which is forming the alpha helical structure of the protein. Okay, length of alpha helix is usually 10 to 15 amino acid residue. There are approximately 10 to 15 amino acid residue joined together with each other. Hydrogen bond forms between the amino group of each amino acid and the carbonyl group of the amino acid 4 residue away. So each hydrogen bond is being formed between one amino acid residue and another amino acid residue, which is 4 amino acid away. So there are different types of bonds that are being produced in between different amino acid residues. Okay, so now what we have to see is how does the reaction takes place, how does this alpha helix getting converted into another helical conformation that is referred to as beta plated sheet. Now we must attempt certain questions which have been asked in CSI and another different entrance examination. One question which was being asked, I'm just sharing that question with you, just let me rub the annotations, probably this is all clear to you. And now see, each and every amino acids is being written as an amino acid residue, then n plus 1 number of amino acid residue. The second is referred to as n plus 2. The third is referred to as n plus 3. The fourth is referred to as n plus 4. And the next one is referred to as n plus 5. So this is how there are n number of amino acid residues that are being joined with regard to each other. Okay. Now, what is going to happen here is that these are different amino acid residues being linked with each other. The first question which was being asked in CSIR, and I'm sharing that the same question with you, is that if there are D amino acid residue, which alpha helix will be stabilized? If there are D amino acid residue, which alpha helix will be stabilized? Can you answer me? You can just write in the chat box. You are given D amino acid residue. Which of the alpha helix will be much more stable? Levorotatory means the left-handed alpha helix or the right-handed alpha helix. Okay, and the next time you are given the L amino acid residue, which is much more stable, the left-handed or the right-handed alpha helix. So what I am trying to speak to you here is that, look here at the note that I am writing. You should have a look at the note. See, if D amino acid residues, if D amino acid residues are given, are taken, if D amino acid residues are taken for alpha helix formation, the left-handed alpha helix, the left-handed alpha helix will be more stable. Okay, now what happens, the next one, the next question, see this, you have to note it down, all these points in your Pathfinder or your notebook. If now the second point, I'm just copying the above one, not to write it out again. So if we are going to take the L amino acid residue, now there are so many intelligent students with us, some may just cut off the first note and then rewrite the second one over it. So do not do such type of silly mistakes. If L amino acid residues are taken, then for alpha helix formation, then the right-handed alpha helix will be much more stable. Then the right-handed alpha helix will be much more stable. But what was the question being asked in IISc PhD entrance examination? The question that was being asked and is also given in the new Pathfinders, that if a mixture of both D and L amino acid residues are being used in poly in polypeptide formation, what would be its conformation? That is, it would be right-handed or left-handed, right-handed or left-handed, handed or left-handed. Please answer. This was the question that was being asked. So the correct answer is that such type of polypeptide cannot assume any conformation. They will be unstable. Okay. Such type of polypeptides cannot be produced. Okay, produced or occupy any specific conformation. When they cannot be produced, then what about the conformation? They cannot be produced, they cannot occupy any particular conformation. Okay, so So that's all. Please do remember this particular point. Okay. Now moving forward, what we have to understand is that now what we have to understand in this particular point is that how these alpha amino acid residues are forming the structure of the protein. Except for amino acid near the end of the alpha helix, all the main carboxyl group and amino groups are hydrogen bonded. Now this question has also been asked that which of the amino groups or the carboxyl group are not hydrogen bonded in the case of alpha helix. The alpha helix except the amino group or the carboxyl group present at the ends of the alpha helix, all others have hydrogen bonding. So the terminal amino or the terminal carboxyl group are never bounded with regard to each other. They do not participate in hydrogen bonding formation. Okay. All the hydrogen bonds lie parallel to the helix axis and points in the same direction. So see this is the helical axis. I'm just trying to draw a helical axis. This would be the helical axis. If this is the helical axis, then there are first amino acid hydrogen bonds and the other pair of hydrogen bonds. Both of these are parallel to the helical axis. So each and every hydrogen bonds always occur parallel to the helical axis in nature. Okay. Next. See, helix can be formed from either D or L amino acid, but a given helix must be composed entirely of D amino acid or L amino acid. Alpha helix cannot be formed from a mixture of copolymer of alpha, D, and L amino acid residues. This question is from IISc Bangalore. Then only we introduced, along with Sir, we were able to introduce this part in this particular book. Okay, now moving forward. See this is the structure. 3.6 number of amino acid residues are present. 0.15 nanometer is the pitch of the 0.54 was the pitch of the helix. And here you can find that with each amino acid that the rise of the helix is 0.15 nanometer. This is the rise of the helix with each amino acid residue. Okay. Now what we have to study is now about the alpha helix. Now see this, there are two important questions which have been asked in CSIR entrance examination from this alpha helix, and that we will have to understand which were those questions. The description of the geometry of the alpha helix. The alpha helix is defined as by the term the pitch. Now this is the pitch by which it has been described. The pitch refers to the distance, the axis between successive lines, the distance along the axis between the successive turns. What is the distance in between the successive turns? That is referred to as pitch. So pitch of the alpha helix is usually 0.54 nanometer or 5.4 angstrom. Okay, the alpha helix has a complete turn of around 360 degree divided by z, delta z divided by p. Now delta z means the distance 0.15, the rise in the helix, and divided by p. P refers to the pitch of the helix. The rise in helix is being determined by the term delta z. Pitch is being given by the term p. R refers to the radius of the helix. Okay. So delta, the change in the conformation of the helix in the 3 dimension. That means change in the angle of the helix is equal to, it is being given by the formula 360 into delta z. Delta z is 0.515 nanometer. That is rise of the helix per atom, as well as divided by the pitch of the helix. There are 4 questions being devoted to this particular formula in the MCQ booklet that has been also shared in the group. Please do solve them up. Okay, thank you. So we are moving forward. Now Nidhi is going to ask something. Nidhi, you can write your question, no problem with that. If this I'm just unmuting you. Okay, Nidhi, you have been unmuted, so probably you can ask the question. Nidhi, you have been unmuted. If you can unmute yourself, you can ask the question, or you can write your question. I will answer it to you. Okay. Okay, there is a query. Can you repeat angular difference again? Okay. Yes, we can repeat the angular difference again. Just a minute. Let me move back. Now see this, when you're going to look at the alpha helix, now there are theoretical questions which are being asked, that I have told you that which are the type of theoretical questions that could be asked. Another way how to introduce this alpha helix is that, now see this, you are living in flat. There is a great flat culture in Bombay and Delhi. So there are flats in which people are living. Okay, and these flats have different types of neighboring molecules. So at a particular flat there is a carboxylic group COOH, and at another there is an amino group, and then there was a hydrogen bonding in between these two groups. Then it leads to the formation of a structure that is referred to as alpha helix. Okay, now is this clear that how this alpha helix was being produced? Now the coming to the formula part. Now what I am going to tell you, फॉर्मूला पार्ट पे आते हैं, फॉर्मूले को देखते हैं कि फॉर्मूला कैसे बना है, how this formula has been formed. Now see this. Let the radius of the alpha helix being r and the rise per residue being delta z. What is rise per residue? I'm just pointing this out. What is this rise per residue? Rise per residue means how much rise has been made on each residue, on each nitrogenous base, how much rise on each amino acid residue, how much rise has been made. Now that we have to understand, you look here, just try to focus on my cursor. You know that the radius could be measured in the form of r, and the rise per residue is equal to delta z. This is delta z, this is delta z. Okay, this is the value of delta z. So what is the value of delta z? 0.15 nanometer rise per residue. Per residue means per atom is 0.15. Okay, so this is delta z, and there is another value that is referred to as pitch. So delta z divided by pitch is equal to change in the angle of the alpha helix. Now supposingly you are standing here, okay, so you are the guy who is standing here. Okay, this is the person who is standing here, and you are the guy who is standing here. What you are going to see is that how much of this revolving ladder is going to rotate with regard to each each atom in the amino acid. How much of this ladder is going to rotate with each atom in the amino acid chain? कितना रोटेट हो रहा है इस सीडी में, कितने डिग्री पे सीडी घूम रही है हर एक अमीनो एसिड के एटम के साथ में. That we have to visualize. With each atom of the amino acid we have to see how much rotation has been made. So there is a formula, change in the rotation equals to change in the rotation per residue divided by pitch. Why pitch has been divided? Pitch is the lateral distance. Now see this, there is a change of 360 degree. Okay, 360 degree into each residue. How many number of residues are being required for a complete 360 degree turn? 3.6 number of amino acid residues are required. So this is 0.15 into 3.6 divided by the pitch, that is 0.54, will give you an angle of 360 degree. Okay, this is what I am trying to explain it to you. मैं यहाँ पर आपको ये एक्सप्लेन करना चाह रहा हूं कि अगर हम डेल्टा z जिसकी वैल्यू 0.15 है, डेल्टा z को बेटा 0.54 से डिवाइड कर दें. डेल्टा z, just do this calculation, 0.15 divide just a second, 0.15 divided by 0.54, then what you are going to get? You are going to get a value that should be multiplied by 3.6, and you will get the number of amino acid residue, how this alpha helix is getting the 360 degree orientation. If you have any query still, you can look at the MCQ questions, and we will discuss the MCQ questions with me. There it would be solved. So this is delta z is rise per helix divided by the value of pitch, and there you get how much rotation is going to take place with each degree. If there is a 12 degree rotation, then how much of the delta z has been covered, accordingly we have to calculate. Okay, now moving forward, so I do think that now it would be clear to you all. Just attempt the MCQs which are given in Pathfinder MCQ booklet. Now moving forward, now there is another conformation that could be achieved, that is referred to as the beta plated sheet structure. Now what is this beta plated sheet structure? That we will have to visualize that how this beta plated sheet structure is being produced. Now please look at the Lippincott. Okay, one more question is there. Got it, sir, thank you, no problem. Now hydrogen bonds. See this. This is what I have told you, that how hydrogen bonds were being produced. Amino acid per turn. Each turn of the alpha helix consists of 3.6 number of amino acid residue per helical turn. Thus, amino acid residue spaced 3 or 4 residue apart in the primary sequence and are speciated close together when folded in the alpha helix. Amino acid that disrupt the alpha helix. Now, there are amino acid that can also disrupt the alpha helix. That is given in the new Pathfinder. Proline is an amino acid that disrupts the alpha helix because its secondary amino group is non-geometrically compatible with the right-handed alpha helix. Now, proline is an amino acid that has an ability to even disrupt the alpha helix. Why? Because their proline has an internal bend within itself. Okay, so proline has an internal bend within itself, so it will disrupt the structure of the alpha helix. Instead, it insert a kink in the alpha helix. Instead, it causes a change in the alpha helix. It introduces a kink in the alpha helix that interferes with the smooth helical structure of the alpha helix. Supposedly, this is the structure of the alpha helix. Okay, supposedly this is the structure of alpha helix. Now, when proline arises, what does proline do? Proline brings a kink. Okay, and because of this kink, for a large area, the alpha helix cannot form a helical conformation because proline itself has a helical structure. This is the structure of proline. Okay, now proline itself has an helical structure. It will not allow the alpha helix to form any helical structure. That is why proline is the one that is going to hinder the formation of bonds in between the two molecules. Okay, so what is going to happen? There is a carboxylic group which is present here. So that carboxylic group was COOH. Okay, there was a carboxylic group COOH. Okay, and there was a amino group in your colony. Okay, no problem. In the building there was a carboxylic group and amino group, and there was a strong type of an interaction which was going on in between them. Then there came the papa of all amino group, okay, in between. Then what is going to happen? All interaction is going to be lost. I don't know who is she. So this is all the interactions are being lost because of the presence of proline in between the two interacting carboxylic group and the amino group. And because of the presence of proline, all the interactions are being lost, and thus it will inhibit the formation of the alpha helical structure. Okay, now see this, hydrogen bonds are being produced. Okay, now finally, amino acid with bulkier side chains such as tryptophan, amino acids such as valine and isoleucine that branches at the beta carbon can interfere with formation of alpha helix if they are present in large number. This is also very important point. So if there are bulkier side chain, amino acids which have a bulkier side chain, if they have a bulkier side chain, then they could also interfere with the alpha helix formation. These questions have been asked. Please note them up. Okay, now we are going to talk about another helical conformation that is referred to as beta plated sheet structure. Up till now, we were talking about only our building, that is a building which is present in helical conformation. Now, do the interactions always occurs within the building? No, my dear, interactions can also occur in front of the buildings. There is one building, there is another building, and then the interactions are taking place. Such type of bonds will lead to the formation of another higher structure which is referred to as the beta sheeted structure. Now, what is this beta plated sheet structure? That we have to visualize. Just wait for a minute. Okay, we are going to talk about the beta plated sheet structure. The beta sheet is another form of the secondary structure in which all the peptide bond components are involved in hydrogen bonding. There are peptide bond components, and these all the different components are involved in the hydrogen bonding. I'm just showing this to you. This is one beta plated sheet structure. This is another beta plated sheet structure. Both the beta plated sheet structure consist of beta strand, and these beta strands can be parallel with regard to each other. They can be anti-parallel. This is anti-parallel. One is moving in this direction, another one is moving in this direction. So these beta plated sheet structure are anti-parallel with regard to each other. Okay, they are both anti-parallel with regard to each other. And another thing could be when two beta plated sheet structures or two beta strands are parallel with regard to each other, so they can assume two different conformation. One is anti-parallel, another one is parallel. Please look at the pathfinder. Now see this. This is very important. There are two different conformations that are possible. One is referred to as the parallel beta plated sheet structure, and another one is the anti-parallel beta plated sheet structure. In the case of parallel beta plated sheet structure, what is going to happen? The carboxylic group and the amino group are going to be present on the same side. For example, look here, there would be a carboxylic group present here, COOH. Okay, sorry, COOH. Carboxyl group is present here. Let me just do this. Okay, this is the carboxyl group which is present, and there is another one, COOH, which is present here. Okay, so there are the carboxyl group which are present there, and here the amino groups will be present, NH2, sorry, NH2 amino groups are present. One amino group is present in here, and there is another group, amino group, which is present there. Okay, NH2. So there are two carboxyl group and there are two amino groups which are present, and both are in opposite both are in the same direction with regard to each other. Then it is referred to as parallel alpha helix. This is also given in the new pathfinder. In the case of parallel alpha helix, full collinear hydrogen bonding is not going to take place. This is the balcony and this is the window. So their full collinear correlation is not being established. Here what is going to happen, because of the anti-parallel strands, because of the two strands which are anti-parallel with regard to each other, full collinear hydrogen bonding is going to take place. This is how full collinear hydrogen bonding is going to take place. Now you tell me in which of these will be stable or this will be stable. The anti-parallel beta plated sheet structure is much more stable. It is much more stable, dear. Why this is much more stable? It is much more stable because it has a bonding present in between the two. Full collinear hydrogen bonding is going to take place. Okay, I'm clearing all the drawings. Those who would have understood, they have already understood what I'm trying to tell. Okay, and those who haven't, they can always ask me again and again. They will never understand. And see this. Here there is the non-full collinear hydrogen bonding, so no interaction, no effective interaction is going to take place. No effective interaction. The effective interactions were present there. Yes, there is an effective interaction. So this is much more stable as compared to the parallel beta plated sheet structure. Each individual strand is referred to as the beta strand. Okay, so every strand which is present is referred to as the beta strand. I'm just clearing all the drawings. Sorry for the interruptions caused. Okay, just a second. I'm clearing all the annotations. Okay, now it's fine. So then what we are going to tell here is that there is full collinear binding, so this is much more stable and this is less stable. Each strand in the case of beta plated sheet structure is referred to as beta strand. Beta strand is an elongated alpha helix. The only difference between the alpha helix and beta strand is that in the case of alpha helix, intra-chain hydrogen bonding takes place. In the case of beta plated sheet structure, inter-chain hydrogen bonding takes place. Okay, this is very important. I'm just writing it out here so that you can understand this particular point. In alpha helix, in alpha helix, intrachain hydrogen bonding was taking place, while in beta plated sheet structure, beta plated sheet, interchain hydrogen bonding occurred. Okay, so there was an interchain hydrogen bonding which was taking place in the case of beta plated sheet structure. So there is an interchain hydrogen bonding that is going to take place. So probably this is clear to you all. Okay, if you have any doubt, you just ask me up. If you have any doubt, you can ask me up. Okay. Rather than being C, beta plated sheet forms two or more polypeptide chain line up side by side. When there are more than two polypeptide chain which are going to lie side by side with regard to each other, then it will lead to the formation of a beta plated sheet structure. Each component of this beta plated sheet structure is referred to as the beta strand. So each of the component is referred to as the beta strand. Rather than being coiled, each beta strand is fully extended. Rather than being coiled, each of the beta strands which are present is fully extended. The distance between adjacent amino acid residue in the case of beta strand is 3.5 angstrom. Okay, the two adjacent amino acid residue are being separated by 3.5 angstrom. Okay, so if there is one amino acid and there is another amino acid which is present below, then both the amino acid will be separated by 3.5 angstrom, and the distance is that of 0.1 is that of 1.5 angstrom. Okay, now the distance in between the two amino acid is approximately 1.5 angstrom, and the two amino acid are being separated by 3.5 angstrom. Okay, in contrast to 1.5 angstrom which was observed in the case of alpha helix. Alpha helix has 1.5 number of angstrom. The two amino acids separated here, they are separated by 3.5. So actually what has been taken place, the alpha helix has been extended by 3 times. Now you look at the camera, that video camera. I'm trying to explain something to you. Okay, and now you look, if this is alpha helix, if this is alpha helix, it has coiled over itself. Okay, it has coiled over itself, then it has led to the formation of this structure, and this structure is that of alpha helix. You take a wire and you just coil that wire over your finger. Okay, okay, when you're going to coil this wire over your finger, then you get a structure like this. Now this, my dear friends, this is alpha helix. Okay, this is how alpha helix is being oriented, that you can see certain structure in the protein data bank that you will observe. Okay, so now this is alpha helix. Now you just take this alpha helix and extend it to a limit of 3 times. Okay, so what was the distance in the case of alpha helix in between two amino acids? It was just 1.5 angstrom. And now what the distance is now, it is approximately equal to 3.5 angstrom. So what has been done here is that we have extrapolated it out. We have extrapolated the alpha helix, and we have extrapolated this alpha helix 3 times. Then it leads to the formation of beta strand. हिंदी में समझे, अल्फा हेलिक्स को 3 गुना और खींच दो तो बीटा स्ट्रैंड बनता है. ठीक है, क्लियर है, यानी कि बीटा स्ट्रैंड ज्यादा एक्सटेंडेड होता है एज कंपेयर टू अल्फा हेलिक्स. So we can conclude that the beta strand is much more extended as compared to the alpha helix. Okay, now stop the video and look at the theory part. Okay, that is given in your book. Now there are two different conformations that it can have. One, it could have the parallel as well as the anti-parallel conformation. There are two different conformations which are being present for the alpha helix. Now, which are these conformations, how these conformations are being arranged, that we have to see. Now, moving forward, these are the alpha helical structures that are present. I have already told you there is the hydrogen bonding between the carboxylic group and the amino group, but not from the same chain. The carboxyl group is different and the amino group is of different chain. Okay, this is not of the same flat. This is of the adjacent flat, adjacent buildings. Okay, there the hydrogen bonding has taken place. Then it is referred to as the beta plated sheet structure. Okay, now moving forward. Now, beta plated sheet structure can also cause the formation of different types of turns. Now, there are questions which are being asked, which is much more stable, anti-parallel beta plated sheet or parallel. Anti-parallel beta plated sheet is much more stable. Look at the Ramachandran's plot. Now let us look at the Ramachandran plot. What I told you yesterday was that the Ramachandran plot could be divided into two different parts. Which are the two different parts? One, it could be referred to as the parallel. Another one could be referred to as the anti-parallel beta plated sheet structure. So here I am dividing the Ramachandran plot into two parts. Okay, this is how it has been divided into two parts. Now, which are the two different components? Let me just write it down. Let me just annotate it out there. There was a parallel beta plated sheet structure and an anti-parallel beta plated sheet structure. So parallel was present in this particular direction. Sorry. Just let me take white color. Okay, so this one was the parallel beta plated sheet structure. Parallel was occupying the less region, and there was another one which was referred to as anti-parallel. Anti-parallel was occupying a very high region in the case of Ramachandran plot, indicating that anti-parallel is much more stable as compared to the parallel beta plated sheet structure. Okay, so there are anti-parallel is the above one is referred. What has been done here? Sorry for this annotation. Annotation has moved. Sorry. So this is anti-parallel beta plated sheet structure, which is much more stable here as compared to this region, which is less stable. Okay, this is the alpha helical structure, right-handed, occupying much more region as compared to the left-handed, occupy less region, found in the case of prokaryotes, in the case of bacterial cells. Any other query that you have, you can always ask me. Okay, even after the class, you can ask me up. Now, there are other different conformations that are possible. One is referred to as the turn, another one is referred to as the tertiary and the quaternary structure of protein. So, but we have to firstly look at Lippincott so that we can do some of the questions that have been asked from the beta plated sheet structure. Now, the beta sheet is another form of secondary structure in which all the peptide bonds components are involved in hydrogen bonding. So all the atoms are being involved in the hydrogen bonding except the terminal ones. So there is, when you compare the beta plated sheet and the alpha helix, unlike alpha helix, beta sheet are composed of two or more polypeptide chains that I have already told you. There are hydrogen bonds which are being produced. Now, the hydrogen bonds are interchanged. This is very important point. The hydrogen bonds which are being produced in the case of beta plated sheet structure are interchanged in nature. In globular protein, beta sheets always have a right-handed curl or twist when viewed along the polypeptide backbone. There can be twisted beta plated sheet structure. Now see this. The alpha helix and beta structure provide maximum hydrogen bonding for peptide bond component within the interior of polypeptide. So these are the ones that provide the maximum. These are the ones that provide the maximum hydrogen bondings. These are the ones that provide the maximum hydrogen bonding. That is why both alpha helix and beta plated sheet structures also traverse the plasma membrane. A very important point I'm noting, I'm providing a note here. Just a second. I'm providing a note here. Note, please note this down. This is very important. Note. Color. Just wait for a second. Here, selecting the appropriate color. Note. Alpha and beta pleated structures occurs in the interior of the polypeptides or those that transverse through the plasma membrane of the cell, that transfers through the plasma membrane of the cell as all amino acids in their structure have hydrogen bonding. Now all the amino acids which are present, all these amino acids have hydrogen bonding in their structure. Because all of these amino acids have hydrogen bonding in their structures, so these are the ones that are ideal for the formation of the alpha helix and the beta plated sheet structure in the polypeptide. Okay, they are the ones which are ideal for the formation of the alpha helix and the beta plated sheet structure. Even certain proteins which are found in the plasma membrane. Now I'm making a cell. Now supposingly there is a cell, and now this cell has a plasma membrane. Okay, I'm making this cell through a two layered, because there is the plasma membrane. This is the plasma membrane. Now there are protein structures which forms transmembrane protein. The part of the protein that is transmembrane will always have either alpha helix or beta plated sheet structure, because both these alpha helix and beta plated sheet structure forms hydrogen bonding interchain or intra chain hydrogen bonding with regard to each other, and so stabilizes the structure of protein. हिंदी में समझ लीजिए, देखिए कोई भी प्रोटीन अगर कभी भी ट्रांसमेम्ब्रेन होगी, यानी कि पूरी मेम्ब्रेन को पार कर रही है, तो उसके इंटरनल स्ट्रक्चर में हमेशा क्या होगा, अल्फा हेलिक्स और बीटा प्लेटेड शीट विल बी present. So this is very important. Okay, so with this we have finished alpha helix, we have finished beta plated sheet structure. I would be informing you about this turns and bands in my next lecture, only because the time is almost over. I cannot finish this topic today by itself. So that's all for today. We will meet at the same time at 8 o'clock. First, I will have my class. Then KK sir, Krishna sir will be having his own class. And so you will have two classes tomorrow. Mine will be shorter and it would be from 7:30. And then it would be KK sir class from 8:30. Thank you. Thanks a lot. Good night. You will get the recording version of this lecture on our official YouTube channel. Thank you, students. Any other query, you can always ask me up in the group or here also.